Hi, good evening, everybody. Thanks for coming in person. Really appreciate. This is another lecture of our C-Secret series. As you know, we are in our 27th season of C-Secret, and this is, I believe, our fourth lecture of the year. You know, with the COVID, we have been operating for a couple of years remotely. We hope that gradually people are going to be coming back in person. It's much nicer to be in the auditorium with everybody. We recommend wearing masks, okay, especially for people that are more sensitive. And it's not mandatory, but it's recommended. And what we request, however, is not to have any food in the auditorium during the lecture. We are thankful to the institutions and organizations and the private people that are supporting this activity. Again, as I mentioned, it has been 27 years, and it's done, honestly, thanks to the donation that we are receiving to be able to continue making it. And a very special thanks to the presenting sponsor, which is the Bank of America. And we have also the Shepard Broad Foundation. We have Bill Galway, Cheryl Gold, the Key Biscayne Community Foundation, the KB Life Enhancement Forum, the John Micon Family Foundation, the William McKehan Trust, and Nicole and Myron Wang that are supporting this specific lecture. So thank you, everybody. Anybody interested to join that very nice group of donors, please contact Jennifer. I'll give the information in a few minutes to participate. We have a tradition at part of this series to introduce as a preliminary speaker one of our alumni from the school that describe what she or he is doing, and you can have an idea of the type of students that we are producing as a school, a great type of students that we are producing at the school. And this evening, I'm very happy to introduce Corina Antal. And Corina is originally from Romania. She grew up in Chicago. She's completed a Bachelor of Marine Science, Marine Atmospheric Science at the Rosenstiel School, but she got interested into the field of marine science as part of an aquatic science course that she took at the Shedd Aquarium. And Corina graduated summa cum laude in 2009 with a double major in marine science and biology. She then obtained a Ph.D. in biomedical sciences in 2015 from the University of California in San Diego. Her thesis work landed her on the Forbes 30 Under 30 in Science list and the TEDx talk. Corina is now a postdoctoral fellow at the Salk Institute for Biological Studies in San Diego, where she is working on identifying better therapeutic targets for pancreatic cancer. Corina will be sharing her research and experience at the Rosenstiel School via a pre-recorded video, and let's take a look at it.
Thank you for that kind introduction. It's an honor to be back in Miami, albeit virtually. I had an amazing time at the Rosenstiel School while I was there pursuing my bachelor's degree in marine science and biology. I was fortunate to have inspiring professors as well as many exciting opportunities that greatly influenced my career path. The Rosenstiel School is a unique place where you can pursue many passions both inside and outside the classroom. For my first semester there, I was exposed to the amazing hands-on fieldwork experiences that marked my time at Rosenstiel. For example, I took part in a saltwater semester, which uses research-based learning to expose students to all aspects of fieldwork. I also immersed myself in the many amazing extracurricular activities that the school has to offer, such as the many great clubs. And no, I'm not talking about the dance clubs on South Beach. I was a part of the Row Marine and Atmospheric Honor Society that promotes awareness and conservation of marine and terrestrial environments. I was fortunate to be able to go on many educational field trips with them. I was involved with the Marine Mammal Rescue Team and enjoyed the amazing diving the Keys has to offer through the UM Scuba Club. I even started a water polo club team my first semester there. The Rosenstiel School has many excellent exchange programs. I was lucky enough to study abroad at James Cook University in Australia, where I got to dive in the Great Barrier Reef and learn a few things along the way while I was there too. But the programs that likely had the biggest impact on my career were my volunteering experiences. I got to tag along with Ph.D. students and assist with their field research. Looking back on it, I was just providing free labor, but at the time, those opportunities made me feel extremely privileged to be able to witness research being done in real time. For example, I took part in the South Florida Certain Shark Program. I always assumed I was one of the lucky ones that got to jump in the ocean at night to hold sharks, but as it turns out, not too many people were keen on doing that. Learning about shark behavior on those trips was a really cool experience that won me bragging rights for years to come. Another great experience was my senior thesis research with Dr. Crawford on the long-term effects of low oxygen on fish. This work not only resulted in a publication, but also sparked my interest in the field of molecular biology, and ultimately is what pushed me to pursue a PhD in biomedical sciences. In trying to avoid a cold, I found my way at the University of California, San Diego. My PhD work there overturned a longstanding dogma in the field that explained why so many cancer clinical trials had failed. From there, I moved across the street to the Salk Institute for Biological Studies as a postdoctoral fellow, and I still am there today. My current research focuses on identifying novel therapeutic strategies for pancreatic cancer. This deadly disease is predicted to become the second leading cause of cancer-related death in the US this decade. You've probably heard of the central dogma where cells use their DNA to make RNA, which then gets translated into proteins. Well, cancer cells need a lot of proteins to grow and divide. So what I found was a particular pathway that cancer cells hijack in order to increase their protein production, and showed that drugs that impact this pathway could reduce tumor growth. Hopefully, these findings will one day translate into the clinic. On a personal note, I'm trying to pass on my love of the ocean and of science that I gained at the Rosenstiel School onto my twins. And so far, I'd say it's going pretty well. Thank you for the opportunity to share a bit about my time at Rosenstiel. And I'd especially like to thank everyone at Rosenstiel for making these experiences possible.
Thanks very much, Corinna. It's great to see you. Unfortunately, not in person, but now I appreciate why it was difficult for you to make the trip back to Miami. Anyway, good luck with your research and good luck with the twins. And hopefully they will become themselves students at Rosenstiel and follow your path one day. You know, we are trying to bring to the school talented students like Corinna. And not everybody has the capability to make it happen in a private school that survive based on tuition. So we are always interested to get support again for scholarships, okay, so that the students can benefit from an education here with us that we love being able to provide. So any of you interested, in fact, to support scholarship for students, please contact Jennifer at the number or at the email that is provided below. It is now my great pleasure to introduce Dr. Bradley Moore, who is a distinguished professor and director of the Center for Marine Biotechnology and Biomedicine at the Scripps Institution of Oceanography and the Skaggs School of Pharmacy and Pharmaceutical Sciences at the University of California in San Diego. Dr. Moore is a biochemist who specializes in reading and writing the genetic code of marine microbes, algae, and sessile animals to develop new ocean-based medicines and molecular tools to treat human diseases. His laboratory pioneered the mining of genomes to discover gene networks that code for natural, bioactive chemicals that support the specialized lifestyles of marine organisms. This work has led to new medicines and new gene tools to monitor oceanic toxins. Dr. Moore has published over 240 peer-reviewed articles and has been recognized by the National Institute of Health, the American Chemical Society, the American Academy of Microbiology, and other scientific societies. Brad, I'm delighted to welcome you to C-Secret.
It's great to be here and be here in person as well. So thanks everyone for showing up. I'd like to, do what I'm going to do here. We're going to talk about reading and writing the genetic code to learn a little bit more about marine life and as well to see how that might impact our own health. That's the goal for today. All right, but before we do that, I want to talk about communication. The communication that, not that the type that I'm using right now to speak with you, but actually the communication that is universal to all life forms here on this slide. How do trees talk to each other? Insects, microbes, the things under the ocean. How do they all talk to each other? When they're hungry, when they need a friend, when they're in danger, or if they're ready to reproduce. What is their language? The language that they use is probably the oldest language that we know on planet Earth, and that's the language of chemistry. I tend to think of small molecules as words. These are the words that organisms use to communicate with each other. We all know these words. We all see this communication happening all around us, whether we recognize it or not. I'll probably give you the one that probably is the easiest for us all to see, where we see organisms that aren't humans or without mouths talking to each other. Ants. We see these in our kitchen. We see them in our yards. We see ants following each other on a single file line, right, to get to something to eat and then back home again. They're all following a chemical that was laid down by the ant in front of it. And we've all done the thing where we've squished an ant and we see them all just scurry around, right? That's a second chemical. It says danger is nearby. There's an alarm going off. In the ocean, anywhere on planet Earth, organisms are using the same trick of using small molecules that only they know and only they can respond to. They've got their ears or their receptors to be able to listen to this chemistry and be able to do this. Here's an example of a coral spawning and all the larvae go out for a swim. Well, these larvae need to be able to settle eventually in a location that's important for it to be able to set up shop and to become an adult larvae. They follow this chemical. It's a very simple organic molecule that's drawn there that's emitted by a bacterium that lives on the surface of mom and dad coral. And they hone that to be able to come home, and then that chemical tells them to settle down and turn into an adult coral. So this chemistry of small molecules is all around us in all life forms, from the microbes to you and me. And in most part, all these molecules are helping all of us navigate our natural world. It's just the way that we look at these molecules and recognizing that there are so many of them, it's very difficult at times to recognize that the sheer breadth of this chemistry that's out there is sometimes hard to really grasp upon. But when we do see it and we understand that it can impact our own lives, many of these specialized molecules, as I call them, have impacted us in many ways. Our own health. There's a picture there of a fungus making penicillin. We've all taken that when we've had an infectious disease. There are hormones up there that help our cells develop as well. So when we think about who that best chemist is really on planet Earth, it's not the guy with his head sticking in the chemical hood there. The best chemist on planet Earth is nature. The guy in the hood, me 30 years ago, is you have to mix together a lot of organic molecules together, often in very controlled environments. A cell gets its instruction from DNA, does it in a very saline, safe environment of a cell, and is able to make the same molecules that we can make as chemists in a factory. So it's pretty remarkable to think that both these events can take place. Now in a cell, and we saw it in the previous presentation, there's something called the central dogma of life. All of information stems and flows from DNA. We know this in Watson and Crick. We understand that DNA is converted into RNA, RNA into a protein, and typically that's where we think of, or we're taught in school often, that that's where information flows. But a protein does a further job. We know them as enzymes, and enzymes do the last step of what we learned from DNA. It converts that message into a molecule. Now, this molecule can allow for information to take place from one organism or one cell to its partner to be able to elicit those types of responses. So we might think of really that molecules, chemicals, have their origin in DNA. We can think of, in us, cholesterol, maybe in your coffee, caffeine. All these molecules, their origin in DNA. DNA is really the recipe of life that gives rise to all organisms around us. All organisms have unique DNA, make unique sets of chemicals that they have, and these molecules we can study in the laboratory. Take a seaweed, I can take some DNA, eventually I can put it into a bacterium, and that bacterium can now begin to make a seaweed chemical. I can bring that back to the laboratory, I can study it, maybe that molecule might have an activity that will impact health, and eventually I can turn microbes into little cellular factories to make small molecules. This is the age we now live in. So let me share a few stories with you where we've been able to do that. Here's a story that starts not too far away from us in Miami, down in the Bahamas. Colleagues of mine at Scripps Oceanography, Paul Jensen and Bill Fennecal, collected in the early 90s a bacterium living in the sediments that has pictured here, makes this molecule on the top left-hand region of this slide. And that molecule was shown to have very important biological properties that somehow told other things to stay away from this bacterium. Don't eat me. Who wants to be eaten? But today that molecule is in clinical trials to treat a very aggressive cancer, brain cancer called glioblastoma. So we all know about different phases of clinical trials now. We've been living in the times of COVID for the past couple of years. So this is in phase 3. So we know this is the last phase before whether FDA approves it or not to become a human drug. So pretty remarkable that this comes from a bacterium living in the sea. It's already treating patients in hospital settings. We wanted to understand how the bacterium makes this molecule. From a practical point of view, we need to be able to learn this so we can be able to envision how to make more of it, make different types, and actually target other types of receptors in the brain. So to do this, we turned to genomics. You can sequence the genome of this bacterium. We did this over 15 years ago. We found over 5 million base pairs of DNA sequence. We can assemble this into a circular map of this circular chromosome of this bacterium. And we can see at that time, this was early days of being able to see islands of genes, sort of in these blue little squares that are at this outermost circle of this genome assembly here. These were all the small molecules that are unique to this and this bacterium alone. These are the words of this bacterium. Two of the molecules that are shown there, the one in clinical trials on the right, and another very powerful agent as well in cancer, are shown there, showing that this bacterium has a lot to say to its neighbors. We can then take this information and we can understand and we can poke and prod it. We can use gene editing technologies to reconfigure how this genome looks to make new chemistry. And on Monday, we're going to publish a paper on being able to understand at granular detail, at the atomic resolution of exactly how this molecule is made inside the cell of a bacterium to make this cancer agent. And this precision of information, of understanding at the molecular level how atoms are moving and breaking, now teaches us and gives us a recipe of how we can accelerate future discoveries of reprogramming these organisms, these bacteria, to making new chemical agents to treat cancer. Today, we've isolated thousands of bacteria and sequenced thousands of genomes. We can take these bacteria, and this video here shows you the rapidness of being able to sequence DNA. This is a new type of sequencing technique. You can see a grad student pipetting into this little device here. This device in her hands is the size of plugs into a USB chip on the side of your computer. This then allows her to actually go in and begin to sequence DNA with a click of a button, and all the cells light up. She's sequencing DNA right now. You don't need these big machines anymore. These are small devices that I can put on the side of a computer. I can do this in a laboratory. I can go out into the field and sequence DNA in real time. Exciting times these are to be able to bring this technology to the problems that we have here. Now, the majority of the sequences that we have are mostly from bacteria. Bacteria have the smallest genomes. They were the easiest to sequence years ago. And so most of the information we have is how we connect a specialized chemical to DNA is really from the bacterial or the microbial world. This latest technology now is accelerating us being able to go after other life forms as well. Plants, animals, fungi, and everything in the ocean. So these are exciting times as well to be able to begin to look at the conversations that other organisms are having. What chemicals are they using for their specialized discussions? And can we in society intercept these chemistries and use them for our own benefits? So let me share with you another example, not from a bacterium this time, but this time from a seaweed. Let me introduce you to a couple of seaweeds that have been known and used for centuries in Japan and elsewhere in Asia to treat certain kind of ailments, especially those for ancient populations that had roundworm or types of infestations. And so these seaweeds, you would actually go and you would collect them, you would bring them home and make a tea out of them, and then drink this tea. And the types of molecules are shown right there, and there's some Japanese, there's a manuscript here on the bottom right showing some of the details that they were using to collect and look for how these molecules were working in populations. We were intrigued to see how we could use DNA sequencing for a non-model organism like a seaweed that someone could collect from us in Japan, ship to us in San Diego, and we could then sequence. So here's a piece of seaweed that was collected for us. We could isolate DNA from this organism, and this organism was known to make this molecule called kainic acid. It's a very small molecule, looks like an amino acid. It actually masks one of our amino acids like glutamic acid, which is an important signaling agent that we have in our own biology. And actually, we have a receptor in our brains called the kainate receptor, named after this marine chemical from seaweeds. And this is an important receptor in you and I that's important for cellular communication, how our neurons are being able to fire responsibly. We could take DNA out of this piece of seaweed. We could then sequence it and sequence another bacterium, another seaweed, and isolate the DNA sequence out of this. Thousands and thousands of genes we could identify, and there were a couple of genes that we were looking for and we identified for the first time. And these 2 genes out of many thousands we're able to take. We can then drop those genes or transplant those genes into a bacterium in the laboratory, and that bacterium now has been reprogrammed to make this seaweed chemical. We can now make large amounts of the seaweed chemical, and this is a chemical that is widely used no longer as a medicine for humans, but is probably used by tens of thousands of laboratories across the world as a basic biological tool to be able to understand neuronal type communication. This is technology we've been able to patent. We can license it as a university, and so forth. In addition to this molecule that one seaweed makes, it turns out nature has been using this kind of chemistry in different organisms, whether it be seaweeds, phytoplankton or diatoms in the middle, and on the right, terrestrial fungi or mushrooms. These molecules all have the same sort of lower half that are identical, and the top half of the molecule is a little bit different. And that top half really tells the molecule how long it's going to park or be resident inside the receptor. And if it has a very low EC50, like the molecule in the middle, domoic acid, it'll stay there for a long time, and it'll cause neurons to misfire disappropriately and so forth. Now, it turns out domoic acid is a toxin. It's a toxin that was first discovered, I think in the 80s or 90s, and a number of people died in Canada after eating seafood contaminated with this toxin. And that toxin came down to be associated with phytoplankton associated with it. A few years ago, in 2015 now, we had on the west coast of the United States, we had the largest oceanic bloom ever recorded on planet Earth. This was a large bloom of the toxic, harmful algal species called Stenichia, which makes this toxin. It got into our seafood, and it shut down a number of our fisheries, including a lot of the Dungeness crab and so forth. That one fishery alone lost $100 million. Beaches were closed to fishing and catch for some beaches for half a year up to a year. We were interested to understand how we could predict these blooming events. I guess down here in Florida, we can predict and track hurricanes that are happening. How do we look at sort of a large event in the ocean that's going to be making a toxin? How can I be able to use the tools of genomics to be able to guide when a bloom is going to happen, whether that bloom is going to be toxic or not toxic? So we wanted to open up the genomes, find genes for making domoic acid for the first time in a phytoplankton. To do that, we went fishing. We went down to Monterey Bay, California, dropped a net into the water. You can fish out all the phytoplankton. You look for the glass rods that look like they're in there with a little micropipetter. You can suck them up, put them in a petri dish, and then start growing those in the laboratory. You can extract DNA, begin to sequence, put that all together, and eventually be able to find what we found here, a few genes out of about 40,000 genes in total. So it's this needle in the haystack we're looking for. We were able to identify the genes, turn those genes back on to be able to make the toxin in a test tube, but we understood exactly what genes are responsible for making the toxin. This then now has allowed us to be able to go into the field and use this technology by putting ocean-going robots in the water through blooms to sniff out these organisms and exactly the genes in these organisms that are being upregulated or turned on to be able to make the toxin. We can then look at what are the physical conditions of the ocean environment at that time that is causing this organism to go from a non-toxic to a toxic stage and then collapse from a toxic stage into a non-toxic stage. So these are exciting times to be able to use this technology, not just to be able to make medicines, but also to look for environmental toxins. The same types of technologies are being used. There's another organism that I want to share with you. I already talked about microbes. We talked about algae. How about animals? Animals have more complex genomes, but they also have a lot to say. And one animal that I have a lot of interest in are those in coral reefs. So we've all been on coral reefs, or if we haven't, we've seen movies, and these are often the most colorful organisms that we see. They're often well protected, and they use small molecules to have organisms not chew on them, or organisms foul them as well. So these small molecule chemicals that octocorals are using are often very well known to the scientific community. But for many, many years, for decades it appears, most animal chemicals have been associated or be thought to be made by not themselves, but actually by the microbes or the microalgae that live in association with them. That's despite thousands of these very powerful biochemicals being well known from corals. These are 3 coral species, one from not too far away from here on the bottom left that makes a really important anti-inflammatory agent. This is a molecule called pseudotericin, was discovered in the 80s by a colleague of mine, Bill Fennicle at Scripps. And that molecule was shown to be a really remarkable, have anti-inflammatory activities. And very soon thereafter was licensed by the university to Estee Lauder, who put that into a cream of theirs to help with reduce inflammation or with wrinkles and so forth, using the natural healing powers of this marine soft coral organism. This opened up whole new industries for people in the Caribbean, and it turned out you can begin to farm these wild corals and be able to clip them and be able to bring them in and be able to make these molecules by stably harvesting these animals in the ocean. We've been thinking, however, that perhaps these molecules may have a different origin, not in their microbes. And today, with fast forwarding to be able to sequence genomes, we can do just that. So sequencing genomes of corals has very recently allowed us to discover a whole branch of genes that discovered for the first time in corals that are responsible for making coral chemicals that have remarkable properties, which are unknown for the animal as of yet, but well known in humans to having activities for curing cancers and other types of human ailments. We could take these coral genes, we can begin to turn them on in bacteria, and they show that they can make a whole health of a wide diversity of coral chemicals that are unique to corals themselves. What we've been finding really exciting about this latest discovery is that these chemicals encoded by these genes are really, really old. They predate similar type of chemistry that's well known in plants. And for most of the plants that we know, when we walk through the forest, we can smell pine trees, we can smell the menthol and others in many of the herbs that we enjoy. These very same chemicals are being made by coral animals, but the corals have been doing it for over 100 million years, longer than plants have. We just, again, don't understand why these corals are making them. And so this opens up a whole new branch of science. So we're working now to be able to repurpose these DNA in the laboratory. And again, by treading lightly on our environment, taking small pieces of an animal to be able to grab a piece of DNA that forever in eternity I'd be able to have that sequence. We can be able to take ships and go down into the depths and look kilometers down and be able to find the hidden treasures of coral chemistry and others that are down there, that are teaching us new ways that these organisms are making their chemistries that might have impact on us and impact in their own environments. So the oceans cover a large part of our planet Earth. much of that, we don't have a good sense of the organisms that live there and really how they're all communicating to each other. We know what's happening. We know they're using small molecules. And I think today, with the advent of all of this wonderful, powerful technology of doing sequencing of genomes, coupled together with being able to edit genomes, write DNA, and be able to take small molecules into the clinic is a very exciting time to be. Now, for me and my family, we all like to go into the ocean, and this is a laguna which is near our home. And so I think not just for wherever you might go to find where you are in the environment to see sort of how you come across different organisms that see each other. I think these types of ways to be able to see organisms, be intimately associated, bring your children, bring your mothers-in-laws out there, and be able to get connected with your environments. All these organisms are seeing each other and talking, and hopefully you'll be able to play in the ocean and play outside as well, and have a deeper appreciation for the different communication that's going all around us when we step outside. So I'd like to end there, acknowledge my remarkable team of scientists at Scripps Oceanography, our collaborators from across the world, and all the funding agencies that support some of the work that I was able to share with you this evening. Thank you.
Thanks, Brad. Really appreciate it. It was fantastic. And Jennifer is going to lead the question section of the part of the talk, okay, if you are willing to answer some of the questions.
Of course.
So if you have a question here in the audience, just raise your hand and we have some volunteers who will be bringing around the microphone. We'll also be taking some questions off of Zoom. So if you have any questions and you're out there in Zoom, just put it in the chat box and I'll get to it in a moment.
So, in fact, I'll start with the first question. You indicated that, kind of the oldest way of communication is through chemistry. And you emphasize that that's true for everybody, but we as humans are using kind of a different way now to communicate. Instead of using the chemistry, we use the physics, of waves that are propagating. Why did we need to make that evolution?
Yeah, it's a good question, but remember, Ronnie, we do use chemistry to communicate. We have all of our senses of taste, of smell. All these are connected to chemistry. You know when to eat certain things and when not to. Your cells are communicating whether you know it or not, and they're using small molecules to do that. And they're not just communicating amongst themselves. Your microbes that live in your gut are actually doing important chemistry for our health. Not just making vitamins, but actually making important neurochemicals that go from the gut all the way to the brain. So we are a vessel that's really connected through all our cells communicating within each other to do chemistry. Of course, when I'm speaking with you like this, I don't have to send you a chemical signal to say, Ronnie, watch out, she's going to get you behind you. She might. But but if I can't move and I don't have a voice and I'm a tree or I'm a coral at the bottom, I've got to be able to do that. And you'll see a lot of insects then will come to rescue or help out a tree if it needs help, if something else is attacking it or so forth. So that chemistry is all around us.
We have another question from the audience. Thank you.
Hi, my name is Nicholas Combs Jesse. First off, great talk. For someone who didn't study molecular biology, it's pretty crazy to me that you can look at a plant and study their molecular biology and then somehow connect that back to some form of treatment for something in one way or another. I was just wondering if you could elaborate sort of on how that process works, because to think that you can study something and then connect it to something totally different is kind of, it's crazy.
Yeah, it is. And for this talk, I sort of, cut a few corners, to be able to get there to share a few stories with you. So there's a lot of ways to do it. Maybe if it's a plant, it might have been a plant that was, either eaten or it was maybe known to make a toxin in it because there was some kind of, maybe we're cavemen and, and our buddy Ronnie took a bite of that thing and didn't feel too good later on. So we knew that was something we wanted to avoid. Or maybe we saw something else, and like in the case of the seaweed, ancient medicine, it was known that when certain flies laid their eggs in that seaweed, they died. And so they thought, well, this would be perhaps something, if I've got an infection, I would take this chemical or take this seaweed. So, okay, so you can connect that from an organism then to that phenotype, I guess I would say, right? But then how do you go from there to the DNA? So this requires a lot of machine learning today to be able to sequence these DNA and be able to understand sort of what all the genes that are there. Typically, we run against the problem where many of them come down to having unknown function 1, unknown function 2. But there are different tricks that you can do to begin to sort of get a sense of the kind of maybe enzyme that gene or protein that it might encode, and that will then lead you down this road. And so a lot of these stories don't take days, they take years to be able to do. And I have the luxury of, in 30 minutes, probably talking about 100 years worth of work out of the lab by many, many investigators in the laboratory.
Well, this is more than a little bit off subject, but I'm curious about your thoughts on the contamination of our marine environment by the pesticides and organicides and fertilizers that we pour into the water directly or indirectly, and also the so-called, what do they call them, permanent molecules like
these
subparts of
persistent organic
Teflon
pollutants,
or eternal molecules. And they, of course, aren't really eternal, but they're going to accumulate and accumulate and accumulate, and it looks like a road to disaster to a neophyte like me.
It's, so it's not a question at all off topic. I think it's remarkably point on topic, because if you think about communication, if it's quiet right now, hey, I can have a quiet conversation with you. If it's noisy, I've got to raise my voice, right, to be able to get my message across to you. So if I think about in the marine environment, if there's a lot of chemicals out there, it's noisy. And I'm having a hard time being able to communicate with you to give that message that through time, we've been already have this conversation for millennia. So having all these, man-made or anthropogenic chemicals in our environment makes it more difficult for nature to be able to have effective communications. And we see that. We see the disease that's happening, right? This is miscommunication that's taking place. I showed you at the very beginning this little chemical about the corals, right? This little bacteria makes this molecule, 4 bromines on 4 carbons on a little ring with a nitrogen plucked in there as well. Very simple organic molecule. Only one bacterium makes that. It's a pretty rare chemical. And so that bacterium and the coral have a really connected, communication. However, if we use salt water in our sanitation systems, our heme proteins from human waste, gets degraded. There's bromine in the salt water, and what does it get turned into? That exact molecule. So there's a group of scientists in Israel that have been able to show that the outflow of a lot of sewer plants discharges that chemical, and if it happens to be in a coral environment, it can perturb these kind of communications. So that's a molecule that has a very short half-life. You also mentioned that there are others that have much longer ones. When you think of DDT or something like this, they just take a lot longer to degrade. So I think, one other thing to say lastly on this topic is we can learn a lot, however, from nature. Nature does a great job at building complex organic molecules. She also does a great job at degrading complex organic molecules. And so I think being able to bring those two worlds together to use, I think, the knowledge of evolution of degrading chemistry that happens in cells all the time to be able to treat, some of these man-made chemicals is within our grasp as well. So I seem to be pretty optimistic about it. Hopefully you are too. Hey, there's only one place to live, and that's in the land of optimism.
We have some more questions here from the audience. I have several online for you. So, anybody? All right, I'll go ahead and take some from the Zoom world. Is there somewhere that we can learn more about your research? Also, hello from the Galapagos.
Ah. Well, hello back at you. So thanks for the question. Thanks to everyone on Zoom joining us this evening in Florida. My research is all public. It's all out there. So if someone were to sort of search for my name under public databases, all of our research is openly deposited. All of our data is openly shared. If we go to Scripps Oceanography, I have a web page, and you can be able to see what we're doing there as well. So we tend to open and release all the data that we have as well. So thank you for that question.
I have lots of them. How do you determine which enzymes or chemicals to do a deeper dive on? Does a cursory look at an organism typically result in an aha moment for what chemicals will be useful?
Yeah, so, you know, I could probably have 5 different answers for this, but maybe I'll just pick one. Usually, often it's driven by a biological interaction. You know, so an organism is doing something. It's either, you know, communicating somehow with a partner, and sometimes otherwise there's a biological observation in the wild of an organism having a trait about it that suggests that there must be a chemical that's facilitating this communication. So then we will use that as a starting point to do a so-called deeper dive to be able to do that. Because, you know, when you think about communication between organisms, it's no different, in essence, between communication between cells. And when we think about when we get sick and we go and we have we see a physician and we have a medicine that we're going to be taking, we take a medicine to actually have an effect on a cell type to do something to it, to stop that cell, to make more of that cell, to be able to stop a certain process that's taking place. That's communication. So that we can see sort of how then chemical communication between organisms is the same thing that's happening in our own bodies when we take a drug. And I tend to think, you know, that since these are evolved types of communications, since nature has been at this job of doing this for millennia versus mankind for just a few hundred years, probably stick with nature. And turns out more than half of our drugs that get from pharmacies are either based directly on natural chemicals or have been modified a little bit so that they can work better in the human body.
The cancer treatment that you spoke of, was it generated by using and studying all of this algae and organisms you mentioned through the portable sequencer?
It's a portable sequencer, yeah. So a lot of those stories at the very end with the algae, with the corals, this is all happening with the portable sequencer, for sure. The beginning stories, we're using older technologies that we had in the early 2000s. These were, again, you know, larger machines as we go from, you know, the technologies of large computers to little computers that are fit in our pockets. The same thing's happening for DNA sequencing technologies. Big things fitting in rooms, costing hundreds of thousands of dollars, to smaller things that cost just a few thousand.
So following on that, someone's asking, what is the name of the sequencing device that you attach to the computer? How do you insert the molecule into it to be able to sequence it?
Yeah, so this is technology by Oxford Nanopore Technologies. They are out of the UK. You can, in the video that I showed, you isolate DNA like you would normally isolate DNA in a laboratory, and then that solution is put into the device. And there are gamillions of pores that are in this device. And each of these pores has the ability to thread DNA into the pore, and it goes through, and each of these pores is then reading a single strand of DNA going through. Not just one pore, not just one strand, one pore, but there are thousands and thousands of pores that are in this. So simultaneously, you're reading lots of DNA. That's why all these little green lights were blinking. Each of these are pores that are functioning, and if you ever had a green light, it means DNA is going through.
A lot of great questions here. So hopefully I can get to all of them. 1 second. Someone is asking about the sustainability of the removal of the organisms. It wasn't very clear.
Sure. So, so that's also, I think, a very important question. And I apologize for not spending a little bit more time on that. As we go from, where this field of science used to be, the field of science used to be where scientists would go out into the field, whether it be, in the rainforest, the bottom of the sea, and you would collect an organism. And typically, you had to collect a lot of it to be able to bring it back into a laboratory. You'd be able to extract out your chemical. Then that chemical you would then test against, different cell lines, cancer cell lines, bacterial cell lines, fungal cell lines, you name it, to find a drug. Today, but on that for one more second, that can be very destructive to an environment because of the amount of material you're removing, especially if it's something rare. Today, if you go into that same environment, I just need just a few centimeters of whatever that sample is to bring back into the laboratory, because all I need is the DNA. Once I have the DNA and I sequence it, then that sequence lives in my computer and in the cloud, for forever. So I have that piece. There are companies now where you can send a piece of DNA on your computer to a company and say, I want this strand of nucleotides. They will then synthesize it at their company, and literally days later, you'll get almost Amazon will deliver this to your doorstep in your laboratory, and you've got that physical piece of DNA that used to live on your computer. I can drop that into a bacterium and put it into the genome of the bacterium, and now it's fixed there, and now making that chemical that used to be being made by whatever it was, down in the ocean or at the top of a mountain. I think that is remarkable sustainability. But this also gets us to the point of, maybe back to your point as well, of we have to take care of our natural environment. Nature's been here for a long time and has evolved these remarkable conversations. Once man-made chemicals get into the environment and they perturb these and we see loss of species, I think that is a loss to mankind, right? That knowledge that has been there and built up through a long time, is going to be lost forever. So I think being able to at least get recorded, DNA samples out there allows us to at least have this. So if we do lose species, then we have that knowledge that's there and use it in the future.
We had so many good questions, and I'm scrolling through here. I want to make sure I didn't ask it already. Did I ask you the question about cancer treatment that you spoke of, generated by using the study of all the algae and organisms
Yeah, we did that
mentioned
one.
through the portal? Okay, thank you. Someone is asking, we'll make this one our last question because we're right near the end here. What opportunities does the seaweed and microalgae chemistry offer to entrepreneurs, producers in the aquaculture industry?
Oh, I think a lot. There are, I guess we think only about the seaweed, area. There is one seaweed there's a lot of seaweeds that people are very interested in making through aquaculture for food, for medicine, for materials, new plastics, being able to make them for feed additives as well. There's a seaweed called Asparagopsis that makes a chemical that actually suppresses methane emissions by livestock, enough that it can be like 50% reduction of emissions. So it can be quite substantial. So there's a budding now field to be able to use aquaculture to generate Asparagopsis to add as a feed additive to livestock to reduce methane emissions, which can have dramatic impacts on our climate.
Thank you so much. That was wonderful. A lot of great questions. Thank you, everybody, for putting things out in the chat that we could read. I'm sorry I didn't get to every one of them, but maybe somebody could reach out to you. If they still have this question that didn't get answered, they may be able to just email you.
Absolutely.
So if you'd like to share this video with your friends that might not have been able to participate tonight, it'll be in the coming days on our YouTube channel, and we'll be posting it on Facebook as well. So one last thing, if you'd like to learn more about our mission in supporting the Rosenstiel School, you can check us out at rsmas.miami.edu. Be sure to join us on April 15th for our Sea Secrets lecture with the Rosenstiel professor Vade Sharaya on revealing the ocean deep next generation sensing technologies for marine and planetary science. Thank you for everyone for your joining us this evening, and have a good evening.